What are EMG&EP? There are techniques that help us to record and analyze electrical signals from our neuromuscular system, including spinal cord and brain. Now, a bit reminder, a bit of anatomy. Here you can see a slice of a spinal cord, and here it is an upper limb of the human body. When I would like to move a limb, electrical signal from the brain goes down to the spinal cord and then through the peripheral motor nerve to the muscle, activate a muscle, the muscle can be contracted. On the other way, if you affect a sensory receptors in skin, this one, they produce electrical signal that goes up to the spinal cord, then up to the brain, and in the brain we can recognize it as sensation. Here it is a muscle and its slice. On the slice of the muscle we can see a lot of muscle fibers. Usually one muscle has up to a few thousand muscle fibers. Here it is the slice of a spinal cord. We can see motor neuron here. One of its axons go through the full length of the nerve to the muscle. It splits into the branches, and this branch with the terminals connects to the muscle fibers. Motor neuron, its axon, and few muscle fibers that are connected to this axon represents motor unit. Motor unit works on the principle all or nothing. And here, between terminal of the nerve and muscle fiber, there is a very small but very strange and very complex part of our neuromuscular system. It is synapse. Here it is. In this picture, you can see nerve motor fibers and muscle fibers. Here it is muscle fibers that will be contracted when muscle contracts. And between the terminal of the nerve fiber and between muscle fiber, we have such a system as synapse. It is a very strange system because it works not on electrical but on base of chemical reaction. Here from the brain, from the motor neuron comes electrical signal. Also, electrical signal will spread. Electrical signal spread through the muscle fiber. But here it is chemical reaction. There are a lot of small bubbles inside the terminal of the nerve. They has a name synaptic vesicles. This bubble contains neurotransmitter fluid. Normally, each terminal has a few thousand such small bubbles. And when electrical signal comes from the brain, these synaptic vesicles go to this synaptic cleft and eject their fluid here. And so the signal can be transferred to the muscle fiber, and muscle fiber can start its contraction. I think this very strange and chemical system is a mistake of the nature. Because in the nature, in some other animals, we can see electrical synapses. That is much more simple. But here in the human body, we have only chemical and very complex. And if it is very complex, sometimes we can get some problems with this synapse. Sometimes this transmission, neuromuscular transmission or neuromuscular junction can be very poor, not reliable. And but if you have EMG machine, it is very easy to examine this system. It's possible to do it, for example, with decrement test. As you remember, during our second webinar, our doctor have shown you this decrement test. Only small reminder. You know that if you apply electrical stimulus here on the motor nerve, the excitation will go to the muscle. Muscle will be activated and contracted because of this stimulus. And we can apply this stimulus few times with some rate, with some frequency, and get a few responses, as you can see in this picture. First response has a very high amplitude, it's a good one. Second is lower, third is lower, and so on, lower and lower. Why are they lower? Because of problem in synapse. Because the neuromuscular junction has low reliability. Maybe not enough synaptic vesicles or some other problems. But sometimes, if you made a decrement test and don't understand, do we have decrement or do we not have decrement? Or maybe sometimes, if it is not possible to make this test, you can use some other technique with the name jitter or single fiber action potential. You can see it here. Here, two single fiber action potentials. Here it is single muscle fibers, and here it is their action potentials. Action potential of one muscle fiber and action potential of the other muscle fiber. How we can record this signal? We put small needle into the muscle near the two neighbor muscle fibers from the same motor unit. And we ask a patient to make small muscle contraction. Our brain sends activating signals. This motor unit will be activated with some rate, and we can record some signals, action potentials. Usually, this delay between two action potentials of the muscle fibers should be constant if you have a good synapse transmission. But if you have some poor neuromuscular junction, some problems with it, you can see some jitter, because the interval between these two action potentials will be sometimes shorter, sometimes longer, and we can see such a jitter. It tells us that we have some problems with the synapse. Reflex Arc. Reflex Arc, it is very interesting system inside the human body. Let's also a small reminder. If we make some stimulus, apply some stimulus to the receptors in the skin, sensory receptors in the skin, they produce electrical signal that goes through the sensory nerve to the spinal cord and then go up to the brain. And in the brain, we can recognize it. On the other way, if you would like to move a limb, our brain sends a signal through the spinal cord here to the motor neuron, then through the motor nerve fiber to the muscle. Muscle will be activated and contracted. But you can see here very strange additional neuron. It is interneuron. As you can see, it connects sensory fibers with the motor fibers and create reflex arc, special loop. How it works? If, for example, you put your finger into the flame, it will be very strange effect, very strange impact, and very big signal will appear here on the sensory receptors. This big signal will go to the spinal cord, go up to the brain, but if it has very high amplitude, it will excite this interneuron. It will be excited, and it in its turn will excite motor neuron, and signal will go to the muscle. Muscle will be contracted, and we withdraw the hand from the flame. It is important to tell you that this system works without our brain. If, for example, this interneuron will be absent here in the spinal cord, how it will work? We put finger in the flame. It produces very high signal. This signal will go to the brain, spinal cord, then up to the brain. In the brain, we will recognize that it is very painful, and brain will send a signal to the muscle through the spinal cord, through this motor nerve. Muscle will be contracted, and we will withdraw the hand from the flame. But it will took too long time. And I'm sure in this, it will be situation minus one finger. It will be too late. So this reflex arc will save our health, and sometimes it save our life because it is very fast, very quick. In the human body, we have a lot of different reflex arcs with 2 neurons, 3 neurons, many neurons. And if you have EMG machine, it's possible to examine such reflexes. Maybe you know H reflex, blink reflex, R3 reflex, and some others. Also, we have such a technique as T reflex that can be done with special tendon hammer, with electronic tendon hammer that is connected to the computer or to the EMG machine, and that is synchronized with an amplifier. When we made a stroke on the tendon, it's synchronized with an amplifier at 0 time, and with small delay, we can get a motor response. Now, a lot about tunnels. Here it is carpal tunnel. You can see in this picture our palm and a slice of a carpal tunnel here. What we can see inside this slice? A lot of part here took carpal bones, because here we have a joint, so they are very big in size. And also we can see here a lot of tendons. Why they are present here? You know, we can move fingers. We can move our fingers, but where are the muscles? Muscles from the fingers are in the forearm. They are far from the fingers, because in the palm there is not enough a place to put muscles here. So fingers are here and muscles are here, far from it. So we have a lot of tendons here. It is ropes that pull our bones, and so we can move fingers. And we have a lot of fingers, and each finger can make a lot of different movements. So there are a lot of tendons here. And you can see a median nerve. Really, there is not enough place for median nerve here. Not enough place. And sometimes because of inflammations, because of trauma and so on, median nerve can be compressed. It can be compressed, and its possibilities for electrical signal conduction will be decreased maybe dramatically, and it start to suffer. And for example, we can see some area of numbness or some other diseases. But if you have EMG machine, it is very easy to examine this tunnel and understand what will be exactly point of the struggle. It is possible to do with such technique as sensory engine. Here you can see we can place recorded electrodes on the finger, on the nerve terminals, and we can apply a few stimulus, put electrical stimulator here, and make few stimulations with only small steps. We only shift electrical stimulator by 1 centimeter. So we can record a lot of waves and understand what is the exactly problem point. Here you can see 6 traces. With first third of them, we can see that the latency of the wave did not change, did not have a big change. But with other 3 responses, we can see that latency changed much more bigger. It is very clear from this graph. First 3 traces and other 3 traces. So it is clear for us that the problem is between third and fourth traces. Very easy. Motor unit number estimation, MUNI techniques. Previously, I've told you that each our muscle have up to few hundred motor units. why it is necessary for us to understand, to estimate the real number of motor units. Let me explain you. Here in the picture, we can see it is schematic picture. We can see two motor neurons that are located in our spinal cord and their axons that goes through the full length of the nerve to the muscles. And here it is muscle fibers. First motor neuron is connected to 3 muscle fibers and second to 2 muscle fibers. It's only schematic. Sometimes we can see a situation when an axon of motor neuron can be damaged. Or for example, motor neuron can be regenerated, can be dry. And in this situation, muscle fibers become denervated. They are not innervated anymore from the axon. They become single, they become alone. And in this situation, if they become alone, they start to send some signal, SOS signal, save our soul signal. It is fibrillations. They become to start fibrillations, and the nearest axon receives these save signals and start to grow a new terminal in this direction of this signal. And this new terminal go to this muscle fiber and create a new synapse. So these muscle fibers will be innervated. This system has a name, terminal sprouting of motor axons. As you can see in the last picture, all muscle fibers are innervated. And if, for example, we would like to record CMAP wave, compound muscle action potential response, or M wave from the muscle, when you stimulate it, we can get a normal response with a big amplitude, normal duration. But sometimes in some diseases, for example, maybe you know ALS, amyotrophic lateral sclerosis, we can get problems with many motor neurons in the spinal cord. A lot of motor neurons can die, can degenerate. But if we record motor response, CMAP, we will get normal amplitude. But in this situation, it is very necessary to understand how many motor neurons are dying, so how many motor units can decreased, and how many motor neurons are alive. And if you have EMG machine, it's possible to do with such a motor unit number estimation techniques. In our system, we have 4 different techniques. One of them here, you can see it is Munix. It will be shown you today. And the last technique I would like to tell you about is somatosensory evoked potentials. On the second webinar, I've told you, maybe you remember, that with the evoked potentials, it's possible to examine a full sensory pathway from the fingers up to the brain. For example, if we stimulate a finger, near nerve terminals, sensory receptors in the skin will produce a signal, electrical signal that will go through the sensory nerve to the spinal cord, then up to the brain. And if we put electrodes on the head, we can record this signal. But the problem is that the amplitude of this signal is very, very low. And it is not possible to see it on the background of EEG activity. But if we make a lot of stimulations, apply a lot of stimuli, and record a lot of traces synchronized with this stimuli, and average these traces, EEG activity will be removed. because it is chaotic. And our response will be very clear visible, as you can see in this picture. Okay, that's all I would like to tell you. And now our Dr. Evgeny Gulaev will show you these expert techniques or some other expert techniques on the real patient. Thank you, and please, Evgeny.